Dear students, today we are discussing about the fetal membranes of chick. So during the development of chick or any other vertebrate, certain specialized embryonic tissue or structures they are produced that may be permanently or temporarily, they do not enter into the formation of embryo themselves. These are external and devoted in one or the other way to care and maintain the developing embryo. These structures are collectively termed as extra-embryonic membranes or fetal membranes or extra-embryonic sacs. These are not the precursors for any of the organs of the adult or larva, but to serve and satisfy the requirements of the embryo in connection with the nutrition, gas exchange, removal or storage of waste materials, and also it provides the protection to the embryo. The extra-embryonic membranes have developed to make the eggs capable of developing on dry land. For example, the eggs of the reptiles, birds and prototherian mammals, they have a protective shell which is present around it. In some reptiles and eutherian mammals, the shell has given way to the uterine development, but the basic form and function of the extra-embryonic membrane has remained the same. Extra-embryonic membrane are those membranes which are formed of embryonic tissues which extend out and beyond the strict confines of the embryonic body, and they are adapted to fulfill the care and maintenance of the developing embryo. So next it is the kinds of extra-embryonic membrane. That means what are all the extra-embryonic membranes you find in the chick. So first one, it is the amnion, yolk sac, allantois, chorion, or you can call it as serosa. So there are four sets of extra-embryonic membrane or the fetal membrane which are present in the chick. So first one, amnion. So this is a very thin membrane which eventually encloses the entire developing embryo in a fluid-filled sac. So yolk sac, it is a most primitive structure which containing the network of blood vessels and encloses the yolk of the egg. Allantois, which serves as an excretory and respiratory structure, it is a large sac-like structure. Last one, that is the chorion or the serosa, is a very thin membrane and covers the embryo and other extra-embryonic membranes like amnion, yolk sac and allantois. It is also fused with fusion of so many amniotic folds over the embryo. So all these extra-embryonic membranes, we call it as a composite structures. That means they involved with the other embryonic membrane as they involve two germ layers too. So the amnion and the chorion, it is made up of, remember, extra-embryonic ectoderm. Ectoderm means the outer membrane and the somatic layer of mesoderm, while the yolk sac and the allantois, they are composed of extra-embryonic endoderm and splenic layer of mesoderm. So in the earlier classes, we have already discussed the mesoderm has been split into two, that is the somatic layer of mesoderm and splenic layer of mesoderm. So here the extra-embryonic membranes, these are the composite structure where they are made up of, that is the amnion and the chorion, it is made up of extra-embryonic ectoderm and somatic layer of mesoderm. Whereas the yolk sac and the allantois, it is composed of two membranes, that is extra-embryonic endoderm and splenic layer of mesoderm. So coming to the development of extra-embryonic membrane. During neurulation, nothing but when there is a formation of neural tube of the chick, The lateral plate mesoderm, they get splits into outer somatic layer of mesoderm at the inner side of the ectoderm, whereas the inner splenic layer of mesoderm, which is lying outer to the endodermal layer. Both these mesodermal layers, they get enclosed in the coelomic space between them. The somatic layer of mesoderm and ectoderm, they are collectively known as somatopleur, whereas the splenic layer of mesoderm along with the endoderm, that will forms, that is splanchopleur. In the picture, you can see clearly what exactly extra-embryonic, that is somatopleur, and extra-embryonic splanchopleur. So at the time of development of the avian blastoderm, the somato and the splenic pleur gradually spread peripherally over the yolk mass, which is far beyond the area where the body of the embryo is taking the form. So here in this picture, you can clearly make out, see the presence of the embryo. Shortly, the embryo proper begins to undercut by the series of body folds that serves to delimit the embryonic regions from more peripheral extra-embryonic somatopleur and the splanchopleur. After the formation of body folds, the somatopleur and splanchopleur of the chick, they start to develop into 4 extra-embryonic membranes, what we call it as amnion, yolk sac, allantois, and the chorion. So the first one, that is the development of yolk sac. So this is the first membrane to grow or to develop, make its appearance. As early blastoderm expands, the extra-embryonic splanchopleur continues to spread over the yolk mass, and eventually it encloses all the yolk completely. It will form the yolk sac. So in this picture, you can see the yellow color, what you can see a bag-like structure, that is the yolk sac. The intra-embryonic splanchopleur is subjected to the superficial body folds, which serves to establish a walled digestive tract, or you can call it as gut, in the body of the embryo. So the middle of the gut, what we call it as the midgut, remains connected with the yolk sac by a narrow stalk, that what we call it as yolk stalk, where the walls of the gut is continuous with that of the walls of the yolk sac. Although the yolk sac is connected with the digestive tract of the yolk stalk, the yolky food reserves are not transmitted to the embryo by this route. Rather, the digestion of the yolk, it is done by the endodermal lining of the yolk sac through the mediation of appropriate enzymes. They will have the digestion. So next, there is a development of amnion and the chorion. So these are the two, they get developed simultaneously, so they will be studied together. So these are both, they are derived from extra-embryonic somatopleur. At about 30 hours of incubation, the head of the embryo sinks somewhat into the yolk. At the same time, the extra-embryonic somatopleur is elevated above the embryo by a folding process consisting essentially of a doubling of somatopleur, which is upon itself. So the initial elevation is over the head end of the embryo, which produce, that is, double somatopleuric hood called cephalic amniotic fold or head fold of the amnion, or you can say it as head fold. As the embryo increases in length, its head starts to grow towards the amniotic fold. As the cephalic amniotic fold gradually extends backwards, so towards the tail region, its caudally, it also start extending, and that what we call it as lateral amniotic folds, which arch over the embryo, and the elevation from the tail region starts, that what we call it as caudal amniotic fold or the tail fold. During the development of amnion and the chorion, you can see the two major development, that is the head fold of the amnion or the tail fold that starts to form during the development of amnion and chorion. All these amniotic folds, they finally converge or they get into at the midline, so that the embryo will encase by two sheets of somatopleure from all the sides except from the region of yolk stalk. The place where these amniotic folds they meet, that what we call it as seroamniotic connection. So this seroamniotic connection opens before the hatching of the embryo and admits the albumin into the amniotic cavity. The fusion of the amniotic folds results in the formation of two sac-like structures or membranes, and also you can call it as the two cavities. So the inner somatopleuric membrane becomes the amnion, what we have discussed, two cavities. The inner one that will becomes the amnion and the outer one will become the chorion, or you can call it as the serosa. The cavity between the amnion and the embryo is called the amniotic cavity, and it will be lined by the ectoderm. So the muscle fibers differentiate within the mesoderm of the amnion, and the amniotic cavity gets start to filled with the fluid, that what we call it as amniotic fluid. The cavity lying between the amnion and the chorion, that what we call it as the chorionic cavity, but it is lined by the mesoderm. So this chorionic cavity is actually the extra-embryonic coelomic cavity, which is continuous with the proper coelomic cavity in the embryo proper. So to summarize the development of amnion and the chorion, you can see first the development of the tail fold and the head fold. Second one, when there is a amniotic fold, they get converge. You can see the two folds, they get meet, that what we call it as seroamniotic connection. So later on there will be a fluid, what we call it as amniotic fluid, and the two cavities will be formed, that is one what we call it as the amnion and the outer one what we call it as the serosa or the chorionic or the chorion. So these are the development what you find during the amnion and the chorion. Next, the last fetal membrane, that is allantois. The allantois first appears late in third day of incubation. It bulges out as a ventral outgrowth of endodermal hindgut, and it consisting of inner layer of endoderm and the outer layer of splenic mesoderm. So the allantois, they are enlarged very rapidly from the 4th day to the 10th day of incubation. So it starts to penetrate into the extra-embryonic coelom, into the space between the yolk sac, amnion and the chorion. Here in this picture you can clearly make out where exactly the allantois have been formed. The base of the allantois there remains connected with the hindgut of the embryo by means of a narrow allantoic stalk. When the body of the embryo they start contract, separating the embryo from the extra-embryonic parts, the allantoic stalk and the stalk of the yolk sac they remain close together, that will form the umbilical cord. The distal part of the allantois penetrates between the amnion and the yolk sac on one side and chorion on the other side. So the mesodermal layer of allantois, they get fused with the other adjacent mesodermal layer of the chorion, where exactly they form a single layer that we call it as chorioallantoic membrane. So when there is a expanding of this chorioallantois, there starts bursts through the vitelline membrane of the egg and pushes the shell outwards. So when they start progressively, it envelopes the albumin and becomes a sac which is filled with the albumin, and that what we call it as the albumin sac that helps in absorption and the water. The allantoic circulation that will function till the hatching of the chick, and when it starts breathing the surrounding air. The umbilical vessels then they get closed. The allantois they dries up and they get separate from the body of the chick. Here is the picture where you can see the development of extra-embryonic membrane in the chick. First, it is the early stage. A picture, you can see the presence of yolk sac, yolk stalk, allantois, and also you can see the caudal amniotic folds and the cephalic amniotic folds. During the later stages, you know that there will be a formation of the amniotic cavity and the chorionic cavity will be formed during later stages. So when you see that is a fully matured stage, that means the allantoic sac, allantoic stalk, albumin sac, vitelline membrane, chorioallantoic membrane, allantoic cavity, allantois, amnion, amniotic cavity, extra-embryonic coelom, and the yolk sac. So these are all the structures you can see when all the fetal membranes have been developed. So this is all about the development of each of the fetal membrane. Starts with the yolk sac and ends up to the allantois. Looking forward, that is the function of extra-embryonic membrane. So extra-embryonic membrane, it is nothing but it should take care and maintain the embryo. For each individual function, there are so many fetal membrane which helps to the embryo. Like that, function of yolk sac, the yolk sac which spreads over the large amount of yolk and serves as a digestive and absorptive organ by which the yolk is made available for the growing embryo. It functions as a first respiratory organ and also it acts as a hemopoietic organ like the liver. The yolk sac also serves as the place of origin of blood cells and at the later stages of development. Here you can see that is the yolk sac. Second one, that is as usual the development, how exactly they takes place together, that is the amnion and the chorion, the functions also goes like that. The amniotic cavity contains a salty fluid surrounding the embryo. Thus, the embryo can accomplish its development in a fluid medium, although which is present on the dry land. Therefore, the amnion serves as a protective organ where the embryo is saved from the danger of desiccation. The amniotic fluid acts as an efficient shock absorber and thus it protects the soft, collapsible and almost skeleton's early chick embryo from mechanical shocks. As the amnion isolates from the embryo, that means from the eggshell, it thus protects it from adhesion to the shell or from friction against it. So the mesoderm of amnion during later developmental stages that will form the muscle cells which contract rhythmically, thus rocking the embryo within the amniotic fluid. This rocking prevents the adhesion of amnion to the different embryonic membrane. It also helps in preventing the stagnation of the blood in the vessels, a condition that might tend to occur on account of pressure from growing organs. The chorion at latest developmental stage joins with the allantois to serve as a nutritional and respiratory organ. Last one, that is a function of allantois. Allantois acts as a reservoir for secretion, that means excretory waste coming from the developing excretory organs. During early stage of development, the chick excretes mostly urea, but later it becomes uric acid. This change is significant as urea is relatively soluble substance and would require large amount of water to keep it at non-toxic level. Uric acid is relatively insoluble and can be stored without any ill effects. The chorion allantoic membrane that will act as a respiratory surface for the embryo. Thus, the yolk sac, amnion, chorion and allantois can be regarded as an adaptation for the egg and the embryo to carry on its development on the dry land. So this is all about the fetal membrane and its development, also that is function of fetal membrane. Thank you.